Okay, so thank you very much, John, for the nice presentation. So as John told you, I'm a postdoc at the CINAC in Madrid, and I'm gonna present you the project that we have just started in collaboration with the Roper Lab at Goethe University in Frankfurt. So as many of you may know, Parkinson's disease is the second most common neurodegenerative disorder, affecting approximately 5 million people worldwide, but it's expected to increase up to 10 million by 2030. Classical symptoms are motor features such as rigidity, tremor, bradykinesia, but also it's accompanied with non-motor symptoms at early and late stages, such as cognitive decline. The two main pathological hallmarks are the loss of dopaminergic neurons in the substantia nigra pars compacta, and of course the aggregation of α-synuclein that will eventually lead to the formation of the Lewy bodies. It is very important to take into consideration that the neurodegeneration within the dopaminergic system is restricted. So it means that it's been reported that there's a preferential loss of the ventrolateral tia compared to of the substantia nigra compared to more medial regions and of the BTA. There are many factors that may explain this selective vulnerability, especially regarding genetic or molecular factors such as calcium handling, oxidative stress, mitochondrial failure, etc. But to the date, there's no consideration of the function that these neurons play and the role that these neurons may have in behavior. So it is important to take into consideration that the brain has two major systems of controlling behavior that are mediated by dopamine, which is the goal-directed system, and the habitual system. The goal-directed system involves voluntary actions, and it engages the associative cortex and the dorsomedial striatum. And on the other hand, the habitual system involves automatic actions, and it engages the sensory motor cortex and the DLS. So as I have already told you, the ventrolateral tia of the substantia nigra projects to the DLS, and it's been shown that in early stages of PD, there's a preferential neuronal loss at this area, and therefore it's leading to dopamine depletion at the DLS. Also, it's been reported that increased corticostriatal activity can be enhanced by a repetitive habitual behavior. This excessive corticostriatal activity increases calcium at the synapses, and it also leads to an increase in the secretion of α-synuclein in the striatal extracellular space that then may be uptaken by these dopaminergic axons. So our hypothesis is that the engagement of the ventrolateral tia of the nigra in habitual behavior can be considered as a new additional vulnerability factor for this selective dopaminergic vulnerability. The habitual behavior will recruit corticostriatal circuits that will be involved in the performance of the automatic task. In our case, we have decided to assess this with the lever press assay. So once this corticostriatal circuit is defined in our specific task, we will also challenge it with injection of α-synuclein fibrils at this point. So here's the methodological strategy that we're gonna follow. So we will target the forelimb motor region of the cortex, especially in the right paw region. We will characterize this circuit, and then we will inject α-synuclein preformed fibrils at these specific coordinates of the cortex. Then we will divide the animals into groups. One will be the control, and the other group will be exposed to enhance habitual behavior by the lever press assay that will specifically affect the right paw. And then we will analyze if indeed α-synuclein fibrils are being taken by the dopaminergic terminals, and of course the influence of this enhanced behavior in the ventrolateral tia of the nigra. So very quickly, as we just have started this project, I will show you the preliminary results that we have obtained so far. So first of all, we have started by the characterization of this corticostriatal circuit involved in the task. So as I told you, we are targeting the four-limb motor region of the cortex and specifically the digit area. So we are focusing on these coordinates. That's why we selected them. And we have performed tracing experiments in order to fully characterize this circuit. So we have started with classic anterograde labeling using FAL, and we have injected these coordinates. Here are the injection sites, and as expected, we've seen that the tracer is being able to transport to the projection, and it does indeed correspond to the DLS region. So in order to confirm this circuit, now we have used retro labeling. So we have used red beads at this, and we have injected them at this defined DLS coordinates. So this is the injection site. And as you can see, we've been able to see the projection at the cortical regions that we are interested, and also at these terminals projections at the level of the nigra. So right now, our ongoing experiments are the important ones. So right now, what we're doing is we're injecting these alpha-synuclein preformed fibrils in these coordinates that we have defined at the cortex, and we are starting with the behavioral assessment of the lever press assay in order to later on study the degeneration that we will have at the substantia nigra. And finally, I would like to thank Professor Roper and Professor Obeso for giving me the opportunity to start this project as my postdoctoral project. To all of you for your attention, and I'm happy to take any questions that you may have, as this is a very starting project, so there's a lot of things that could be asked and we can comment on. Thank you.
Thank you so much, Josefa. So, well, it's early morning, so there are no initial questions, but there might be questions at the end. So I'm going to ask is, so with this kind of mouse model, how much of a delay, like between the control mice and the sham and the Parkinson one, like how much is that motor delay obvious in the behavior, you know? And how much do you think this motor symptom, like, because a lot of my work shows that it's no surprise people with motor disease have a motor issue, but how do you think that'll inform the other symptoms as well by learning, trying to address the motor part of the symptom of the disease? Or like, well,
we are trying to with this model, we are trying to mimic more kind of like the early stages. So we are supposed not to get many motor manifestation. We may eventually have some, but right now what we are seeing with the lever press assay is that there's no difference in the motor behavior of the control group and the group that has been exposed to this lever press assay. So right now, what we're most focused in is that if this enhanced habitual behavior will at the end affect the nigra. So we expect to see at the behavioral level no difference between the two groups. That will be the ideal. Maybe, of course, as we are overstressing them with this task, we may eventually see also some changes in behavior, but our initial thoughts it's that there's supposed to be no difference in the motor.
You know, it's funny you say with some of the stuff we show in Parkinson's is that we show that people with Parkinson's have similar motor response times, but they're doing it by compensating by recruiting more areas. And then when we tax the system, that's when we see the real behavioral differences. But to see the more subtle differences, you have to look neuronally, you have to look at these kind of things. So yeah, and also early stuff is so hard. Parkinson's being so hard to diagnose and symptoms being the primary course. It's so important to understand that early stuff is vital.
Yeah, you know, totally. So this is,
you can live very long without actually realizing that it's the beginning of a Parkinsonian symptom.
Exactly.
So that,
yeah. This project is also focused exactly on that phase, on the prodromal phase. So it's been known that since you start showing symptoms, the neurodegeneration process may have started even to 10 to 12 years before you show any symptom. So we are trying to focus on that period, even though we know that from the patient side, it's still very hard to diagnose that moment. But also, as there are all these biomarkers coming through, we hope that in some years there may be a clue. So that's why we want to focus on that prodromic phase with this project, in order to study more at this moment where we think that therapeutic interventions may have an effect. Because as you said, like when you start showing symptoms, sometimes it's already too late, and any neuroprotective treatment, it would be very hard for it to restore the system back to the beginning. So that's why we are trying to center on this prodromal phase in order to find a therapeutic target that may work at this moment, with the help of all the biomarkers studies going on, in order to be able to translate this into the patients and to be able to give an effective treatment at the right moment. That would be the...
No, it's very true. Like, definitely, because I only do stuff in humans, is that you have someone who's progressed very far in the disease, and still there's subtleties that aren't there, and it's too late. So that is actually a great